Hi, my name is Amanda Kocher, and this is my presentation for action potential here for the first week of Concepts in Human Physiology 1. So I would like to start with a little review of general neuron structure. So we have the dendrites that pull in those incoming nerve impulses, and that's where we see our graded potentials. The cell body, where all of that's interpreted. The axon hillock, which is where we actually get that enough potential to meet that threshold in order to activate our actual action potential down the axon of the neuron, which is where that action potential or that nerve impulse will be propagated through to take it to the axon terminal, which will either take that impulse to another neuron or possibly to a muscle in the case of a neuromuscular junction. So the action potential itself is a nerve impulse that will travel down the axon of the neuron. It is going to actually cause a change in membrane potential that will reverse the polarity of that cell membrane momentarily and essentially create a wave-like impulse down the axon through that electronegativity charge. It does follow the all or none principle, in which once that threshold of negative 50 millivolts is met, the action potential is going to go. So it does not go backwards, it does not think about it. Once it meets that negative 50 millivolts, depolarization is going to occur. On the graph, you might notice those little small spots here where we have those little graded potentials, those little stimuli that started moving our membrane potential toward that threshold, however, did not meet the actual threshold to send it into a full-on action potential. So this differs from those graded potentials that we would have discussed previously regarding actually getting to that threshold. So what goes on during an actual action potential? So this graph is one of those graphs that you have to be able to know and explain through and through when discussing an action potential. So we start out at that area number 1, which is generally the resting membrane potential, right around negative 70 millivolts. Different graded potentials will move through the cell body caused by a stimulus, and these are decremental in the fact that they will kind of spread out and lose some of their oomph as they get there. But if we have potentials that reach the axon hillock and meet that negative 50 millivolt threshold, then the action potential is going to begin down the axon through the rising phase of depolarization. And depolarization occurs when the sodium channels open at that threshold and sodium is allowed to diffuse quickly into the cell, which causes the spike in this graph, moving us through area 2 and takes us to the top of that peak at about positive 30 millivolts. So it's almost a 100 millivolt swing from our resting membrane potential to where depolarization actually ends. So when depolarization actually ends, you enter the falling phase or repolarization. This starts at that same positive 30 millivolts where we ended depolarization, as this is a continuous process that happens within a matter of milliseconds. The sodium channels are going to become inactivated and close, while the potassium channels are going to open and potassium will diffuse out of the cell. This is where students typically get confused in that they try and make one ion positive and the other ion negative, but both ions, sodium and potassium, are positive. Sodium rushes into the cell during depolarization and potassium rushes out of the cell during repolarization. This allows our membrane potential to return to a more negative state, and actually, as that potassium diffuses out of the cell, it is going to kind of overshoot that resting membrane potential and go into a state called hyperpolarization, where it has actually gone past the resting membrane potential to a point where it is very hard to stimulate that section of membrane again. So this is where we enter the recovery phase or refractory period. And that makes it very hard to stimulate that area of neuron for a new action potential, which we'll talk about in just a minute. But as the sodium-potassium pumps do their job in pumping sodium and potassium against their concentration gradients, that's what allows the membrane to return to that resting membrane potential there that you see near number 5 on the graph. That is when this area of membrane is ready to receive a new stimulus and potentially allow for another action potential or nerve impulse to move forward. All right, so that refractory period that we mentioned a minute ago, there are 2 parts of it. So there's the absolute refractory period when that area of membrane is not able to be stimulated at all to propagate an action potential. This would be during that depolarization and repolarization phase, in which that membrane is already undergoing depolarization past its threshold, or it's repolarizing, in which it's already still above that threshold, so it is not able to be stimulated again. The relative refractory period is that time frame when the membrane might be able to be stimulated if there's an exceptionally strong stimulus that is trying to move a nerve impulse forward. However, this is when that area of membrane is hyperpolarized, so it's been moved farther away from that action potential threshold that must be met in order to propagate a new potential. So it has to be a very strong stimulus in order to get that area of membrane to propagate an action potential. So let's talk ion channels. So in order for an action potential to occur, that depolarization and repolarization process has to happen quickly and efficiently. So the sodium channels and the potassium channels are the 2 main ion channels that contribute to this. The sodium channels are going to open their activation gate at that threshold of about 50 millivolts. And this is when cell depolarization can occur, because sodium will flow with its concentration gradient through the membrane into the cell. Through this depolarization process, you have a spike in membrane potential, taking you up to that roughly positive 30 millivolts. And at that point, the sodium inactivation gates close. So you have 2 different gates on your sodium channel that are going to allow for activation and inactivation. Then, once that cell returns to resting membrane potential, then you're going to see kind of a reset to that inactive closed state of those sodium channels. The potassium channels, on the other hand, work kind of opposite. Remember, they work hand in hand. So the potassium channels open their activation gate at that positive 30 millivolt membrane potential, and they will remain open until the membrane potential returns to resting, although it does continue through that hyperpolarization state. So that's why those pumps are so important, to make sure that the sodium and potassium levels are being pumped against their concentration gradient and getting that membrane back to resting potential. The last thing that I want to chat about in this is the concept of saltatory conduction. And so we've talked about this action potential, this wave of spikes that goes down the axon as that nerve impulse is propagated from the cell body to the axon terminals. Myelination is one way that we can speed up that propagation of the nerve impulse along that axon of the neuron. So myelin is composed mostly of lipids, and it wraps in multiple sheets around the axon forming different types of cells depending on which area of the nervous system it is associated with. So in the peripheral nervous system, we see Schwann cells, which is what is shown in the typical neuron drawing on this slide. You see they almost look like little bubbles of myelin. And then you have the nodes of Ranvier in between those myelin Schwann cells. And those are little areas of exposed axon that we'll talk about in just a minute. But I also want to note that the oligodendrocytes are where we see this myelination in the central nervous system. So those nodes of Ranvier, these are what allow for saltatory conduction. So as an action potential is traveling along an axon, you are getting that action potential to basically hop over those Schwann cells or over those myelin-wrapped sections of the axon, which is going to speed up your action potential along that axon through orders of magnitude. It is a very large jump in speed and allows for very quick transmission, even along longer axons, as well as a nerve impulse has to go through several neurons to get where it's going. It just speeds up that process so that you're able to quickly and efficiently react to a stimulus. Thank you very much for listening to this, and I hope it was beneficial.